So this is not a cumulative exam. Whatever labs we have done after the lab midterm, immediately after the lab midterm, all those labs are in this test. Okay. So this test goes in two parts, part A and part B. Why is that? There is a reason behind that. You can understand that when you get the test, so don't need to worry about it. But then why am I mentioning about two parts? Is because part A is something that you will be given at the beginning of the test, and as soon as you get it, you can start taking the test. And after you're done with part A on the Scantron, you're going to turn in only part A test to me, not the Scantron. Keep your Scantron, okay? Give only my test back to me. Then I will give you part B, okay? And then continue the Scantron, the same bubble part, right? Wherever the question ends. The questions end on part A with 40, number 40, okay? And 41 to 60 are on part B. So that's how it's gonna go. So those are some of the things. And coming to the lab, you guys did enzymes lab after your lab midterm. You guys did enzymes lab after your lab midterm. So enzymes lab will be on the test, and then followed by respiration, photosynthesis, and human phenotypes that I made you guys do, mitosis, meiosis. That lab, we all did that lab. That lab will also be there. How about Mendelian genetics? You did not do that corn counting experiment, guys. Lab 12, Mendelian genetics. You did not do that part of the human phenotypes and Mendelian genetics lab 12. Part of it we did not do. Chi-square test and all that we did not do. Okay. So that will not be there on the test. That will not be there on the test. I believe Brittany, Stacy, Tracy, give me a thumbs up. I believe lab 12 is 105 page number. Am I right? Page number 105. Okay. So guys, 105, 106, 107, 108, 109, 110. 105 to 110, you don't have to worry. There is nothing on that in the test. Part of lab 12, which is human phenotype, which is from 111 onwards, I covered that in online sessions. Or in Zoom meetings, Zoom recorded sessions under genetics assignment. Under genetics assignment. That is the lab quiz 12 that you have taken. So that will be there for your final lab exam. Oh, are we going to do genetics problems on the test? Yes. You are going to do genetics problems on the test. Genetics test that you take at home for 50 points, that is open book. That is open book. Okay, Sierra, all your hand movement is making me dizzy. So either put that away or else, yeah, thank you. So human genotype, human phenotypes, genotype, phenotype, and all that. Those genetics problems will be on the test. You should know the concepts on how to do it, and you should be able to do it. Okay, guys? You should know how to do it, and you should be able to do it. There is no help. It's not open book. Your genetics assignment test that you take at home is open book. Homework is all open book, right? And as soon as the students walk in, I want you to hand in your genetics assignment, the work that you have done by hand for taking genetics assignment test. I need you to hand it to me as soon as you walk in. Make sure that you write your name and date at the top right corner. If possible, on every sheet of the genetics problems homework. Without your name and date, I won't be able to give any credit to you. What I'm going to do when you give that to me and go, be seated and do whatever you need to do, wipe off the table, and I do a little preview, a short one because we are doing it already here. And I give you some time to study to see how you are based on my assessment of asking you questions and listening to your answers. I'll give you 5 to 10 minutes to study, maybe not 10 minutes, we will see. And then what I'm going to ask you to do is come to the front of the lab, and I will place you in different order than how you were sitting. And then I'll start the test. There will be some station-based questions just like midterm, but this time it's all multiple choice. This time it's all multiple choice, no written. Okay. And while you guys are doing that, I'm timing you at the table and you guys are taking the test. I'm keeping an eye on you to see whether it's all going well. And at the same time, I'm also grading your genetics assignment just to check if you have done all the 24 problems or not by hand. And then I will grade it to you. I'll enter the grade in the grade book and I will give it back to you when you are leaving. When you are leaving the lab, I will give it back to you. I'm not going to keep it. So don't leave it with me. I want to give it back. That's what I'm trying to say here. Okay. And coming to the questions. Any doubts here? If you raise your hand, virtual hand down there, and when you click on reactions, there is virtual hand. If you raise your hand, I can let you unmute. All okay so far? Yes, Brittany.
Okay, so how are we supposed, can you hear me? Sorry, I have a different headset in. Okay. How are we supposed to know if we're doing the genetics assignment correctly? So that, like, I understand, like, I've watched the videos and I feel like I grasp the concepts, but how are we supposed to really know that we're actually understanding and grasping the concepts without having it graded beforehand?
Most of the questions are self-explanatory.
Okay.
A couple of things that you can do. One is you can go to tutoring center for help. You need not go physically. They are available online also. It is called Smart Tutoring. If you go to Valencia College website, you can book an appointment and they will look at your work. That is one way to do it. Second thing, I have provided guidelines on how to get it correctly. Even in spite of that, you have problem with one or two questions. I don't think every question is an issue, right?
Yeah, I agree.
Some questions are pretty easy based on what I explained and based on the videos that you have seen. Still, you think that you have a problem, then email me the picture of what you have done, the work you have done. I will let you know if it is okay, if it is acceptable, or you need to rework on it. I will not give the answers. Please understand that this is like any other test, but then I can definitely guide you. You're asking for guidance. I will guide you. Hey, looks like there are more Punnett squares required. I don't think you did the percentage correctly. I don't think you started correctly actually, right? So that is where everything fell apart, because if you don't start right, naturally you're not going to get it right. So I'll give you guidance like that if you take pictures of the selective ones and send me by email. But if you send me like 2 hours before the test, not you, Brittany, I'm telling others, because it's being recorded also. If you send me 2 hours before the test and you expect me a night before the test and students expect me to respond to them, I'm sorry, I'm not that great. Okay, I'll try my best. I understand that you have tests tomorrow, so sometimes in the middle of the night I might check and I might get to it. But then there is a high possibility that I might not get to it if you do it last minute. Yeah, I know, Brittany, you don't do that. Just letting you know.
I'm done with mine, so I just don't know if I did everything the right. Sorry.
Oh, definitely. Yeah, we should. I mean,
Okay.
Questions are such that you would know many of them, whether you got it right or not. And some questions you have you have problem understanding the question itself because the way they worded.
Yes.
I see what you're saying.
Okay.
Any
Thank
other
you.
questions, guys? Thank you. No? Guys, there is no point in attending and not turning on your video, honestly. It's totally your wish not to turn on the video, but then not turning on the video is not quite helping you. Let me tell you. Okay? There is very less accountability when you're turning off your video. Yes, Tracy.
Morning, Professor. I have a question. So for the final lab, I checked the lab guide, and for the final lab exam, there are some terminologies like memorized definitions of like item, molecule types of bound. Those actually were from the chapters before midterm. So do we still need to follow this lab guide?
Yes, all of it. I'm going to cover those. That
Okay.
that is actually part of second part B, I said, right? That is actually part B. Okay,
Those concepts.
I'll cover that.
Thank you.
So you take out your lab manual and also a notebook. I cannot guarantee you that I will be going really slow, that you will be able to get everything and repeat everything exactly the same way that I say it, guys. I say it off the top of my memory, however it comes. I just make up a sentence, okay? So I don't remember exactly what I said. If you ask me to repeat after my fifth sentence is done, what was the previous sentence, okay? So please do write down. Writing is going to be very helpful. When I say in my final lab exam review part in the lab guide, I hope you have that as well. Guys, do you have your final lab exam review? Yes, Stacy, that one. I'm so sorry, not lab exam review. I meant to say lab guide review. Lab guide. If you have lab guide, it is really helpful. This is the lab guide. And if you look at the last page, you will see this So you have in labs 8 to 12. Okay. This exam is all multiple choice. It has no short essays and drawings. You don't have to draw. Doesn't mean that you can ignore all the drawing work that I made you do. Quiz questions will also be included in the test. This is exactly what I wanted to have, focused on, guys. So all these you studied for lab quizzes, right? Most enzymes are made of blank. Some enzymes are made of blank. Hydrogen peroxide, what is its molecular formula? What is it broken into? What is it broken by? And height of the blank, what does it mean? I hope you have all the answers. I made the guide key available as well. You have key also. So I hope you have those. Quickly raise your hand, one of you. Virtual hand, not physical hand, virtual hand down there. Give me the 4 factors that affect the rate of enzymatic reaction. Raise a virtual hand down there. I can't see if you're raising only physical hand. Yes, Brittany.
I have temperature, pH, concentration, and reactants.
Concentration of what?
Substrate concentration.
Substrate concentration and
product reactants.
That's not the one. Let me see. Tracy, you 3 were correct, Brittany. Okay.
Substrate concentration, temperature, pH, and absence and presence of enzyme.
No, it's not absence and presence of enzyme. It is absence and presence of activators and inhibitors. Absence and presence of activators and inhibitors. Not letting me find the chat here. Okay. Activators and inhibitors. Hey guys, I provided the key. Are you not able to access? Doesn't that have it? Is this not given in that? This is not given in the key. Okay, gotcha. One of you, Kathleen, Stacy, BB, Sheila, Lulu, Tanisha, Ariana. Okay. Ariana, Sana, Sierra, Sierra. Now, I need one of you to answer the other questions. One or all. Raise the virtual hand, please. Go ahead, Stacy.
I'm sorry, which question? The number, that one?
1 to 4.
Okay.
Any one of them. If you have the key in front of you, you could see the key and answer, right, guys?
I don't have the key in front of me. I have it in the canvas, but I don't have it printed. I don't have the answers printed.
Okay, you raise your hand. So sorry. You want to answer anyone? No? Okay. Go ahead now.
Number 1, most of the enzymes are made of proteins. Some are made of RNA.
Very good. Sheila?
Let's see. Number 2, when hydrogen peroxide is broken by a catalyst, then water and oxygen bubbles evolve.
Okay. What is catalase here?
Just write it in here.
Are you asking me? Sorry.
No, Sheila muted herself.
Oh.
Sheila.
There you go. What was that?
Anybody?
Professor?
What is catalase?
And catalyst is something that is it that breaks up or
Good try. Sierra. What is catalase? It's an open-ended question. There is no specific answer to it. Okay. You can say anything that you remember. Huh.
It's the decomposition of hydrogen peroxide to water and oxygen.
Not quite. Good try.
Do you know the
Sana,
answer?
what is catalase? I know Joanna knows. Sana, you raised your hand. Yes, Joanna.
The catalase is the enzyme
Good. How do you know?
because it ends in ASE.
Exactly. That's a hint. The reason why I asked that question is because there is a hint there. There is no way I can remember all the names of the enzymes that are working in human bodies and animals and plants, right? There is no way. But then there is a clue right there. It's a hint. ASE, the end in ASE. What are the other suffixes that enzymes name end in? Does anyone know? Yes, Joanna.
So it's they end in A's, in, and there's something else.
Zyme.
Zyme.
No worries. One more, couple more people. Now, Sheila, Stacy, Tynesha, go ahead. Any one of the questions, quickly.
Number 4, substrate binds to the active site on an enzyme due to complementary shape and chemical compatibility. The active site is the answer to number 4.
Active site, very good. Okay. Did you all memorize a chemical reaction for cellular respiration correctly? Correctly is the key here. And you should know both the name and the molecular formula. And a chemical reaction for cellular respiration is slightly different from a photosynthesis chemical equation, guys. The products on the chemical reaction of respiration would have ATP. Okay. Tanisha,
Yes,
any
ma'am.
part of lab 9? Anything, any answer to lab 9?
Chemical reaction. I don't know.
Okay.
Oh, no.
Can you all download your key and take 10 minutes to read that, guys? There's no point in me doing the review. Yes, now. Cindy, are you able to hear me? I don't think so.
Hello?
Yeah, yes, Kaylee.
Sorry,
think
glitching and
Cindy
it was
is hearing me.
frozen. Yeah, it's all frozen. Oh,
My screen is frozen?
okay, not anymore. It's not frozen anymore. Sorry. I think it's my internet is really slow.
Okay. Cindy, are you there? No. She's not hearing. She's there, but she's not hearing. Okay. Anyways, Tracy, something that you learned, and then Stacy, and then Sheila. Quickly, what did you learn from the key? Or whatever you remember from the lab, or else if you have any questions. Tracy, go ahead.
From all of them, enzyme, cell respiration, photosynthesis, mitosis,
Anything.
meiosis.
Mm.
Yeah, I learned those subjects and the concepts.
Okay, something particular, like 1 or 2 points.
Particular, the equation and yeah, a lot of things. And enzymes are made of proteins, and some are made of RNA. And photosynthesis is just a reversible reaction from cell respiration. And light is necessary, can be both sunlight or bulb light. And there's another equation about the carbonic acid in the lab involve the
Thank
water and carbon dioxide. Okay, thank you.
That's good. Stacy, something that you learned by doing the labs that you wouldn't have learned otherwise, if there is anything.
Let's see. We did endergonic and exergonic. Endergonic is energy in, exergonic is energy out, which is the heat, and then anabolic and catabolic. In the lab, we talked about kinetic energy with the perfume smell. Was that not, that wouldn't have been 9, that would have been like 8 maybe. So never mind on that one, right?
7. No worries, no worries. Thank you.
That's it. Okay.
That's good, that's good. Brittany, something that you learned by doing labs that you wouldn't have learned otherwise, or something that came as a learning from the lab.
Stacy, were you talking about diffusion? Sorry.
I did. I jumped. Like
Okay.
my brain went, I don't know why I did, but it jumped all the way.
Okay. That's what I was like, so she's talking about diffusion, right? Okay, thanks. I am kind of, there's so much, I feel like I've learned so many things that I wouldn't have. I feel like bringing concepts into like my life, where like learning about how cell energy, and like when I cook in the kitchen now and being like, oh, I know why this happens. And I know why when we break down certain foods that our body can process it easier depending on like the needs of our cells, and being able to like look at, oh, my kid has a fever, but my child has a fever, and I have to make sure that the fever doesn't get too high, because then their enzymes can unravel, and like actually grasping concepts from what we're learning in lab and being able to apply them to my real life and understand how everything works and why it works is just, I wouldn't have been able to put those two things together without the lab.
Good job.
I don't know. Does that answer the question?
Yeah, it was an open-ended question. Toward the lab, what from the beginning till now, what were you able? What did you learn by doing the experiments that you did or activities that you did?
Yeah.
So yeah, that was good. It made more sense, or the concepts in theory you were able to apply.
Mhm.
is what I heard. Lulu, then Sheila, and then we will go on to the concepts in the key. Okay, Lulu, go ahead.
So I learned about diffusion through the membrane, specifically about like hypertonic solution and the hypotonic solution, hyper and hypotonic, where hyper is like more concentration from the outside compared from the inside, and then hypo would be less concentration, solute concentration from the outside compared to the concentration from the inside of the cell.
Perfect, thank you. Sheila, I have a lot to cover, guys. I have to cover the genetics part, I have to cover the mitosis, meiosis part, and the rest of the lab. So go ahead, Sheila.
Well, I learned a lot, trying to scrunch it all in there. But to summarize, I guess, I know how to use the data logger now with photosynthesis and all that. That was fun to do. I didn't know how to do that before.
Yeah.
Mhm.
Yeah. One more.
Let's see. The worms were really interesting to see, the germinated beans too, and ungerminated beans in that lab.
Okay. Good. Okay. That was good to hear, guys, that there was something that you were connected with, or you were able to connect with. Okay. So something that Sheila said, I would really want to highlight that. Okay. It's very, very important. Maybe not for the lab exam, but for the future and for your life. Real quick. Are you all with me? She talked about data logger. She didn't know what is data logger. In that lab, she figured out how to turn it on, how to operate it, how to make it work, and how to get the data out of it, right? Complete juice of it. Something like that you have to do in your entire life. When I came to US, I didn't know how to assemble a desk chest drawer. There we get everything ready-made. We have labor so cheap that if we need anything like that to be done, we call a carpenter and they come and do it, right? We pay the money. But here I have been an electrician, plumber, everything. I watch a YouTube video and I do all the plumbing work and electrician work and many things. Okay. It's not because I'm in US, but I think I would have done that because being a scientist gives you that learning, that anything that you do not know, you can learn. Give yourself some time, practice, do it trial and error multiple times, and then the third or fourth time you will succeed. That you have to do your entire life, especially if you are in sciences. Especially if you are in sciences, let me tell you that this is how the scientist life will be, or biologist life will be, or a healthcare taker life will be. You have to figure out. Okay, figuring out is the phrase. You have to figure out. Okay. So coming to enzymes lab. Everything that is there in that key or the lab guide for the enzymes quiz, that will be there. You have to understand that there is a molecular formula of hydrogen peroxide which is close to water. Please don't mix them up. On the test, you will be in rush to see things. Don't rush. Calm down. Cool off. And make sure that you see correctly what you need to see. That happened a lot. Maybe not with all of you, but it happened a lot. There is a difference between catalyst and catalase. There is a difference between catalyst and catalase. Catalyst is potassium permanganate is also a catalyst. Okay, chloride can be a catalyst, but they are not catalases, right? Catalase is an enzyme. Catalyst is anything, any chemical, any molecule which will enhance, speed up the chemical reaction. Does it only speed up? It's known to only speed up. Okay. If they are made of organic molecules. If they are made of organic molecules, then they are enzymes. If they are made of organic molecules, then they are enzymes. And these organic molecules are of two types, proteins and RNA. Proteins and RNA. Is DNA, are enzymes made of DNA? No, enzymes are not made of DNA. Enzymes are not known to be made of DNA. Enzymes are made of, some are made of RNA. And there is a little confusion here I've seen in your lab quizzes, guys. When I say rate of reaction, means the speed at which it is happening. Please understand that rate of reaction means the speed at which it is happening. Okay. Can it also mean that the slowness at which it is happening? I don't think I've seen a phrase like that in any books. Okay. So when I say rate of reaction is directly proportional to temperature, what I'm asking is, rate of reaction increases with increasing temperature or decreasing temperature, or neither? No. Somebody is answering it, so I was saying no to them. Rate of reaction increases with increasing temperature only to a certain point. If the temperature increases above that point, then the rate of reaction falls. So they don't go parallelly all the way. So rate of reaction is high at an optimal temperature, and rate of reaction is low at a high temperature. Rate of reaction is low again at a lower temperature. This was part of your lecture quiz. If in the human body, the optimum temperature is body temperature. So for human enzymes, optimum temperature is human body temperature. Okay. Whereas for enzymes in bacteria from hot water pools, if you take their enzymes and try to do experiments in lab, then they don't work well at human body temperature. They don't work well at human body temperature. Okay, real quick question. Enzymes in humans work the best at blank temperature. You must know the human body temperature here. Cindy, do you know what is human body temperature? (98.6), 38.6. Yes, Mariana. 98.7 is in Fahrenheit, 38.6 in degrees Celsius. Fahrenheit is never asked in sciences, guys. Never asked in sciences. So if you know it in Fahrenheit, you have to convert it to, at least for the test say, convert it to degrees Celsius. Okay. Let me ask you one more question. Ready? What is the optimum pH for, no, I'm sorry. What is the optimum temperature for enzymes in human stomach? What is the optimum temperature for enzymes in human stomach? Listen to the question. What is the optimum temperature? What is the optimum temperature for enzymes in human stomach? Till the body temperature. So you have to listen to the question carefully. Just because I said stomach, you jump to pH. Though I was saying optimum temperature, right? The reason why I said this question or orally, verbally, is because I want you to know that this is how your brain plays tricks with you guys. You don't read the question. I'm sorry, I don't mean to say this in a harsh way like this, but then I don't know how else to say this. You guys don't read the question correctly. You jump to the conclusion. That is the reason why many times, though you can catch your mistakes, you don't, and you end up losing points. All I'm trying to say is don't do it. Okay? Catch yourself. Yeah? What is the optimum pH for enzymes in human intestine? Put it in the chat. I don't want to repeat the question.
Which part? Which part, Professor? Which body part?
Intestine. Colon, intestines, not colon, intestine.
I see. Okay.
Small intestine and large intestine. Some of you, I got you again. Yes, Mariana. Why did I say yes to Mariana? Because my question was, what is the optimum pH for enzymes in human intestine? Yeah. I know I always repeat, but then I wasn't repeating this time. Yeah. It's actually more, it seems, Tracy. It's more than 5, 6, it seems. It is nearly 8. Really alkaline. Anyways, what I'm trying to say is, thank God my tests are not oral, right? You can read and reread as many number of times as you want, because orally I said it and it's gone. There is no way you can ask me to repeat unless I repeat generally, which I do, which I did not do it for this question. Okay, coming back. What is the factor that increases with increasing rate of enzymatic reaction? That factor is increasing and enzymatic reaction is also increasing. No, sis, not glucose. They're both directly proportional. No, baby. Bubbles are the product. The rate at which the bubbles are forming is what my question is. See, the rate at which bubbles are forming, which is the enzymatic reaction. Enzymatic reaction is increasing with this factor. We talked about 4 factors, right? One of them is directly proportional. The factor is going on increasing. Enzymatic reaction is going on increasing, it seems. It can't be temperature. Don't give all of them. Not all of them, Tracy. Yes, Saraya. Only substrate concentration. Only when substrate concentration is increasing, rate of reaction is also going on increasing with it. I told you just now that temperature, with rising temperature, rate of reaction increases only to a certain level. After that height, after that temperature called optimum temperature, when you start to increase the temperature, rate of reaction falls. Are you okay with 106 degrees Fahrenheit body temperature? Your body temperature goes up to 110 degrees Fahrenheit. Are you okay with that? You're not okay, right? Why? Because your enzymes are unfolding. Unfolding is the word. They degenerate. At higher temperature, egg becomes omelette. And you can't get a chicken out of omelette. I gave that example not because I wanted to crack a joke in the class, but there is a reason behind that. At least in the form of a joke, you will learn. So with increasing temperature, rate of reaction only goes up to a certain extent. So rate of reaction and temperature are not directly proportional. Are not. We can't say that. Are they inversely proportional? No, they are not. Then how does that go? It is parabola. It is bell curve. It is bell curve. Okay. You should know what are inhibitors and what are activators. What are enzymes made of is proteins, and proteins are made of amino acids, and between amino acids, what is the name of the bond? This is part of your homework question. Make sure that you not only study lab guide part, but you also study the end of the lab questions in the lab manual. Okay, here in the lab manual. I'm not talking about the end of the chapter questions in textbook. I'm talking about end of the lab questions. Make sure you do those because this is one of that questions. We did an experiment with yeast cells and red blood cells, right? I took a drop of blood and added what on top of it? Can you go to that experiment? Now, Sierra, Sana, Ariana, or Deterica, can one of you answer this question? We took a drop of blood and what did we add on top? Let them answer, Stacy.
Was it sodium chloride?
No, sweetie.
Peroxide.
Can you look at your lab manual, please? Yes, Sana. What peroxide? Give me the complete word.
Hydrogen peroxide.
Hydrogen peroxide. We added hydrogen peroxide on top of it. What did you see on adding hydrogen peroxide? Look at your experiment, guys. Give me a second, please. Okay, I'm back. When you pour hydrogen peroxide on yeast cells and red blood cells, what comes out? I think somebody answered bubbles. You see bubbles. But then for bubbles to come, you need to have one more ingredient. What is that, Brittany?
A catalyst?
Catalase. Where is catalase here? I added blood and hydrogen peroxide. So where is catalase?
The catalase would be the hydrogen peroxide. No?
Catalase, and hydrogen peroxide is hydrogen peroxide. They're separate, they're different, right? But where is this catalase? When you added hydrogen peroxide to yeast cells and red blood cells, you saw bubbles.
Okay.
So where is catalase?
In the enzyme?
Where is
No.
the enzyme? Catalase is an enzyme. Where is the enzyme?
In the blood?
In the blood. In the blood cells. Your blood cells contain catalase. Don't think that, just like nowadays kids think that vegetables come from the backside of the Walmart, right? They think that the storage of the Walmart creates vegetables, right? Magically. Like that, don't think like that. The catalase is actually what cells make from genes. We have genes to make those proteins. That is why I said catalase is a protein. Enzymes are mostly proteins. What difference did you notice between the germinating beans and the non-germinating beans? In their graphs, I mean to say. It was part of cellular respiration lab. Yes, in cellular respiration lab, you used two, which produced carbon dioxide, Brittany.
The worms, I think, produce carbon dioxide, and then the beans were
Which
oxygen.
beans? I'm saying there are two beans, right? Germinated beans and ungerminated beans. What is the difference between them?
The germinated beans, because they were the green part, like the green part.
They are not green at all.
Oh, sorry.
There's nothing green in it. Sorry, it's okay. There is nothing green in beans, right? I'm talking about cellular respiration lab only. I'm asking you, the question is, what is the difference between germinated beans and non-germinated beans based on the experiments you conducted?
Non-germinated bean is dead bean, so it doesn't have any reaction. So the graph will be like straight line for both oxygen and carbon dioxide. But it's different for the germinating beans. It's gonna have cellular respiration reaction. So the oxygen will decrease. At the meantime, carbon dioxide will be increasing during the reaction.
Exactly, right? Think of you as germinated beans. If you were put in that chamber, then you would keep taking in oxygen. So amount of oxygen reduces and carbon dioxide increases in the chamber. You are exactly like germinated beans. Okay. Somebody is asking for something, guys. Give me a second, please. Okay. I texted. WhatsApp. Sorry, guys. Okay. In cellular respiration, on earthworm, you did the experiment to check with a data logger and oxygen sensor and carbon dioxide sensor both, which is Vernier gas sensor, whether they release those gases or absorb those gases, and based on that you were able to conclude whether they are carrying out cellular respiration or not. Okay, so that was an experiment, but the question is, which part of their body are they taking in oxygen and releasing carbon dioxide from? Yes, Tracy.
Professor, can you repeat the question one more time, please?
Which part of their body did they use to absorb oxygen and release carbon dioxide from?
Each cell does it.
Which part of their body?
Body, everywhere. Whenever there's a cell, cellular respiration happens, and I think through cell membrane.
Not cell membrane, it's skin.
Oh, skin.
Skin, guys.
Okay.
It's the skin surface, surface of their body.
Okay, so the question is about the organs. Okay, skin.
Okay.
Mm-hmm.
I hope you wrote that down, guys. And there are questions about which instrument is used for measuring the amount of oxygen, and what was the function of data logger. They were all questions from lab quiz as well. And so here is a quick question which you might have not thought of. When the amount of light increases, does photosynthesis increase or decrease? We are in the next lab, okay? We are not going to talk much about respiration now. This is about next lab, photosynthesis lab. Amount of light increasing and rate of photosynthesis is increasing. Are they directly proportional is a question. Probably your experiment has given this answer, or maybe it did not give the answer. We don't know. Vivi says yes. As the distance between the leaf and the light is decreasing, the rate of photosynthesis is increasing, right? More light, more photosynthesis. Distance is decreasing, that means light increasing, right? Amount or intensity of light is increasing. I said it in many different ways. I hope you wrote it down. And when you read, make sure that you apply what I'm trying to say here. What was the name of the little instrument that you used to measure the intensity of light? Light sensor, very good. And what is the unit of measurement for light sensor, light measurement? Very good, Stacy. No, not parts per million. Parts per million is for oxygen and carbon dioxide, okay? For light measurement, there was a different unit of measurement. Lumi, no. Lux. Lux is the unit of measurement for light. I'll be changing some questions from the test. I'll ask some of the questions not to do on the test, and I'll give instant questions on the board right then and there. Okay, depending on how you're answering on the preview. Okay. Why do I do that? Because there are two things. One is commonly the entire class is good with some concepts, and I want to also test who has really studied. Okay. Probably I'm talking in jargon, but that's okay. That's not an important thing. Let's move on. Carbon dioxide is absorbed in dash process, blank process. Carbon dioxide gas is absorbed in which process? Good job, Stacy. Stacy is talking in codes nowadays. Yes, baby. Okay. Is oxygen released in photosynthesis? Is oxygen released in photosynthesis? Oh my God, unanimously. You see how some questions are easy for the entire class, and some questions people give me answers in codes. I have to sit and decode. Okay. I hope you remember that phenol red is originally what pH? Okay, in words. Good. And as soon as it mixes with a blank gas, it changes to acidic. Stacy wrote it in the chat, Tracy. It's neutral. pH is originally neutral and is red in color. It mixes with which gas? Not carbon. Carbon is not a gas. Carbon is an element. Phenol red mixes with which gas and changes to acidic? Yeah. Mariana gave the answer. Good. Some of you are giving the answer. Next one. That experiment where I blew bubbles into phenol red, right? There was an experiment where I blew bubbles into phenol red. What was the purpose of that experiment? What was it trying to prove? See the lab exercise. I'll wait. This session is not to test you and ask you questions and torture you, asking you to give answers. This session is to learn. Carbon dioxide is not acidic. Sorry. Good try. Good attempt. Go ahead, BB. You can unmute.
So you're asking when you blew the bubbles into the phenol red? That's what you're asking?
Yeah, that experiment, what was the purpose? What were we trying to prove with that experiment?
Oh, you're showing that when you blew the bubbles, carbon dioxide gas was being mixed and changing the color, forming carbonic acid?
That was for that activity specifically. Yes, Brittany, 1 second. But the whole entire experiment, what was that experiment for? What were we trying to prove? What was the objective of that experiment? What was the objective of that experiment? Yes, Brittany, go on.
It was to prove that photosynthesis works, that they remove carbon dioxide, that plants remove carbon dioxide with light. Because you put the plant inside and then the light came and then the liquid, the phenol red, turned red again. Like the
Exactly. Go on, finish.
No, I think that was it.
Okay, so that experiment, the objective of that experiment was to prove that plants absorb carbon dioxide during photosynthesis. Plants absorb absorb carbon dioxide during photosynthesis. I'm looking at so many plants here. Can I say that they're taking in carbon dioxide? Is there any way I can prove it in the air? I can't, right? So I take phenol red, I blow bubbles, I know that I'm releasing carbon dioxide because that is what we exhale. And then you saw visually that it changed from red to yellow. How? You know that you are blowing carbon dioxide. When carbon dioxide mixes with phenol red, it changes to yellow color. You saw that instantly, right in front of you. And you kept the plant and you let it carry out photosynthesis because you put light on it. And then there you go, it carried out photosynthesis, and during photosynthesis, it separated carbon dioxide from carbonic acid, and it ate up all of that. Because it carried out photosynthesis, when they are carrying out photosynthesis, they absorb carbon dioxide. So this was the entire thing. So like this, every experiment, you should be able to absorb it into your mind, guys. That is the point of doing science classes. If you just formality's sake, you just go do the lab and whatever they are saying, do and come back and don't understand anything, don't think about it. Why did we do it? What was the purpose of this experiment? What did we try to prove? I did not understand. I need to go back and ask. If you don't do all this, it's waste of your time. You'll finish the entire undergraduation, you'll finish the entire PhD also, and people don't have anything in their top floor. Okay, let's go to mitosis, meiosis. We all know PP, MATC, right? We all know haploid, diploid. Do we know what is haploid, diploid? Yeah. And do you know the difference between monohybrid cross and dihybrid cross? What is gamete, right? Let me see. Let me ask some of these questions. Easy one, okay? Anybody can answer. Put it in the chat. Where do you find chromosomes in metaphase? Where do you find chromosomes in metaphase?
Mitosis or meiosis?
All. They are there in mitosis, meiosis 1 and meiosis 2. Let me specify the question. Where do you find chromosomes in metaphase inside a cell? What is the location? Many of you gave the answers. Not on the cleavage furrow. At the metaphase plate, middle of the cell, on the middle line. On the middle line, these are all the answers. Center is a point, middle line is a line. So I would, instead of saying center is not wrong, but not correct either. So not center, you will say middle line. Okay. Yeah, exactly, Stacy. I want you to put in the chat, where do you find chromosomes in prophase? In the nucleus, where? In the nucleus, everywhere. In the nucleus, everywhere, right? Not nuclear envelope. In the nucleus, everywhere. In the middle, in the center of the cell, center area of the cell, everywhere. Where are the chromosomes in anaphase? There is no nucleolus in prometaphase, right? Nucleolus is almost disintegrating in prophase, actually. Little away from the middle line. Just slightly away from the middle line is anaphase. They're pulled away from the middle line. Yes. How about telophase then? Telophase and cytokinesis. Where are the chromosomes located in a cell in telophase and cytokinesis? This is the easiest part of the test, guys. The hardest part is what is coming up. It's 11:28. In fact, you know what, guys? I think I'll take a short break if you don't mind, please. I do need a break. I'd go without any breaks in the lab. I prefer breaks in lecture classes. I pause recording. So A positive is a phenotype, AB positive is a phenotype. For A positive phenotype, there are 4 possible genotypes. For AB positive phenotype, there are 2 possible genotypes. The question is, when you cross or one parent is AB positive, the other parent is O negative, can they have A negative child? Go ahead, Aminda, click on the share screen at the bottom. You can select whiteboard and do it on the board and tell me why if a parent is AB positive and the other parent is O negative, they can have a negative child. Tracy, why don't you do it? I don't know if she's able to do it or not. At the bottom of the Zoom screen, click on share screen and then you will have a bunch of choices. Select whiteboard. You should be able to do whiteboard. Are you able to do? BB, you want to give it a try? Oh, no, not on this, on the whiteboard. Can you do whiteboard? Brittany is able to do it. Go ahead, Brittany. Wow, I like your straight line. You see now what kind of masterpieces I was drawing? You have no idea how much it took. Good job. You're drawing all the gametes. In O negative parent, that's the only gamete possible, O negative. That's all. That's enough. You already got A negative there, right? So yes, they can have A negative child. So next question, would you please stop sharing? You may stop sharing.
I don't know how to stop sharing.
It's right there somewhere. It says stop sharing.
Fudge. Oh, I don't know how to go.
There is a red box. It says stop sharing.
I see it.
Yeah, good. Okay.
Sorry.
Here is a question. The parent is B positive. A parent is B positive. Other parent is O positive. One parent is B positive. The other parent is O positive. Positive, positive. B positive, O positive. First question. Can they have a O negative child? They can. Yes. Yes, they can. Can they have O positive child? Yes. Can they have B negative child? Why not? They can have B negative child. So what? I said what? B negative or B positive earlier? Unmute. You can unmute, all of you.
Be positive.
B positive. So B plus.
And O plus.
There's no B. Right. B plus O plus B negative O negative. Okay, and did I say the other parent is O positive?
Yes.
So for O positive parent, it is O plus is one gamete. Other gamete is O negative. So what did I say? Can they have O negative child is what was my question, or B negative?
You said O negative?
This is O negative, right?
Yes.
And this one is B negative. This one is B positive, this is O positive, this is B positive, this is O positive, this is B positive.
So the answer is yes.
You have to know what gametes are possible, and you have to pick the gametes from which there is a possibility and rule out. Last question. Last question. AB positive parent and O negative parent. AB positive parent and O negative parent. Can they have all children? Otherwise, let me ask you this way. AB positive parent, O negative parent, can they get a O negative child?
Yes.
No.
No. They cannot get a O negative child. Because one parent will give either A or B. No parent will give O. And to get a O, both parents should give O. So if a parent is A, B, other parent is O, they cannot have O in the next generation whatsoever. None of their children will be O. None of their children can be O. Well, how am I doing it in the head as if, we are all professionals? Many of you are able to do. It's not just me. Many of you, you did the homework, you know the concepts, you are able to do. Others, you are not able to do. It is not a huge mistake. It's no loss. All I'm trying to tell you here is, guys, you have 1 week time. You need to sit and study. Please make sure that use this 1 week time, certain days. I don't know how many ever number of days you can possibly study. You need to really invest in studying. That's all. Others did. Many of the people in the class did. BB did. Tracy did. Brittany did. Joanna jumped out. But you were not even required to attend this class, Brittany. So this was extra for you. Okay, guys. That's all. That's all from me. Unless you have any specific questions that you want me to answer. Oh yeah, I forgot. You want me to answer those lab guide questions, right? What are the definitions of all those words? I'll give the definitions right now. Atom is a fundamental unit of matter. Molecule is a combination of 2 or more atoms of same element or different elements bonded by covalent bond or ionic bond only. Types of bonds. There are 4 types of bonds given in your textbook. Covalent bond, ionic bond, hydrogen bond, and van der Waals interaction. Covalent bond is due to sharing of pairs of valence electrons. Ionic bond is due to transfer of valence electrons. It need not be pairs. And cation is formed by losing electrons. Anion is formed by an atom when it gains electrons. Isotopes and isomers. Isotopes are atoms of same element with different number of neutrons and different mass numbers, but same number of protons, same number of electrons, same number of same atomic number. Who is cute? Brittany's son? I don't know. Tracy's son. Tracy, that's your son behind? I don't know. I couldn't see. Sorry, your screen got this.
He left. Yeah.
He left. Okay. So he came like full moon and left. Adorable. Okay. Coming back to definition of a cell. Cell is the fundamental unit of structure and function of an organism. It could be plants, animals, humans. Humans are also animals, bacteria, but not viruses. Organelles. Organelles are the closed structures inside living cell, eukaryotic living cells, made of membrane, plasma membrane, inside which there is space to carry out metabolic chemical reactions. Such structures are called organelles. Bacteria do not have organelles. Viruses are not even organisms, so they do not have organelles. Cellular respiration is a process, metabolic process, wherein any kind of food molecule, nutrition molecule is broken down with the help of oxygen or without the help of oxygen into carbon dioxide and waters or hydrogen sulfide or hydrogen carbonate, I believe, in order to release energy from the nutrition molecule. It starts in cytoplasm and most of it takes place in mitochondria. Photosynthesis is a process wherein ingredients like carbon dioxide and water are taken in, combined with the help of sun's energy trapped into them, with the help of molecules in chloroplast to produce nutrition molecules. Mitosis is a cell division where the number of chromosomes at the beginning of the cell division and at the end of the cell division in each cell are same. Diploid number is maintained. Meiosis takes place in only mature reproductive organ cells in order to produce sex cells, wherein the number of chromosomes become half. They go from diploid to haploid at the end of meiosis. DNA replication is copying of a chromosome and making two sister chromatids, which takes place in the interface of a cell or interface of a cell before mitosis and before meiosis during the S subphase. After DNA replication, the copies of the chromosomes are still attached to each other from end to end, but they're closely attached at the centromere. Transcription is a process which takes place in all cells all the time, whether they are undergoing cell division or not, wherein a gene is copied and gene which is in the form of a DNA is copied and the mRNA is produced. Copy of a gene in the form of RNA molecule is produced. Sometimes it is ribosomal RNA, sometimes it is mRNA, other times it is tRNA, transfer RNA. Translation is using the information on the copy of the chromosome, which is messenger RNA, and putting together amino acids and synthesizing a polypeptide. Transcription and translation together make gene expression or protein synthesis. Gene is a sequence of nucleotides found on a chromosome. A chromosome is a structure inside the nucleus. It is there always. During prophase, it starts to condense, and during pro-metaphase and metaphase, it has condensed completely. So you see it as a darkened structure under the microscope. Genetics is the study of genes and gene transfer from one generation to another, wherein we study any deformities or abnormalities and mutations also. Bioinformatics is a marriage between biology and computer applications, wherein we use computer applications to process biological data. Biotechnology is a marriage between biology and technology, wherein we use instruments like data logger that you used to measure the data in biological organisms. It could be in bacteria, plant cells, cancer cells, animal cells, people. For example, your PET scanners are biotechnology equipment. Microbiology is a study of microorganisms which cause diseases. It is not study of diseases, it is study of microorganisms. That's all, guys. The reason why I did that in one go without any pause, without looking at you or asking you whether you got it or not, is because all these are definitions, basic definitions from your textbooks. Many of them are from lab manual. You have been doing all this from the beginning of the semester. I need you to go over all of this and take this all with you when you go to the next semester. Just because you have passed this class doesn't mean that you can put all this in a bag and throw it on an attic and forget about it. That's not what sciences are. Sciences are basic courses that prepare you to put your feet on and jump up. Go higher, go higher. Your basics should be strong. If your basics are not strong, anybody can come and play with you and you won't even know, and we sound like dumb fools. Just because we are not MIT graduates, Harvard graduates, Cambridge graduates, doesn't mean that we shouldn't know our basics. If you know your basics, you are equivalent to Harvard graduates. Okay? And that is my wish for all of you. Make sure that you know what you should know and then go forward. Don't try to just get grades. And I always insisted whenever you came to my class, don't come to my class only for attendance. I don't like it. If you are here with me, then you paid me. You better get the work out of it, right? That's all, unless you have any questions.
Professor, I have a question.
Yes.
Okay, I have some questions about the three types of cell mitosis. We know you told us skin cell is lifelong mitosis. And
Not
muscle
all
cell.
cells, Tracy, right? Only 4 or 5 types of cells only carry out mitosis all life.
Okay.
Only 5 types of cells. Underneath skin cells, nail bed cells, and hair cells, that hair follicles, and mucosa, which is not top layer but underneath layer, because top layer is dead, and bone marrow. These are the only cells that carry out mitosis lifelong. Whereas your muscle cells, bone cells, nervous system cells, they divide only till you are 18 if you're a girl and 21 if you're a boy, and after that they say stop, no more, you can't grow any more taller.
And liver, right? Is liver the only organ that?
Liver is the only organ. If it is scarred, then it will come back to cell division. It gets its cell division capability. That's why even though the person, even though liver donors have only one liver, when they get operated, they say that I'm able to donate, I will donate. Then they operate on that person. They will take only a part of their liver and give it to the person who needs liver, and their liver who gave liver, part of their liver also will grow back. The new liver that was given to the recipient, that liver also is going to grow and give them full liver.
How about blood cell? Yeah, how about blood cell?
Blood cells are mature cells, like your top layer of skin cells. They live for 25 days and they degrade. They're gone.
So they divide it by themselves, or the bone marrow?
Bone marrow. Bone marrow will carry out mitosis and will add continuously every day.
So blood cell did do cell division? No. Oh, that's
bone marrow cells do. Bone marrow cells do.
So
blood cells don't do typically, but white blood cells which are cancerous, they divide, and that is called blood cancer. white blood cells which lost capacity to stop cell division, they divide in blood. That is what is causing blood cancer.
Leukemia.
Normal blood cells do not divide. Normal blood cells do not divide. Blood cells which lost control over their cell division divide, and they cause blood cancer, leukemia.
Thank you.
Blood cells, normal blood cells don't divide. Abnormal blood cells divide. Any more? Any other questions, ladies? Yes, Brittany. Go ahead, unmute.
Is there a Zoom meeting tomorrow too, or is it canceled because school's closed?
No, Valencia has Thanksgiving break from tomorrow to Sunday.
Okay, so then we'll have class, like Zoom class again next Wednesday.
Hmm.
Okay.
Yeah.
That's it.
Our lab is Tuesday though, correct? This Tuesday coming up is our lab exam.
Yeah, most of the people in today's Zoom session, you have a class, you have a lab, in-person lab next Tuesday. Come to lab to take the lab exam and say tata bye-bye to me.
And bring our homework.
I have
And
a
bring
weird
your homework.
I have a weird question. Tracy, Stacy, or Mariana, are you guys getting together and doing a study session that I could join? Because
I'm happy to.
You would? Okay. I don't know how to, do you want me to just text you my number?
Sure.
Okay. Or I don't care. I don't care if anybody texts me.
Why not, right?
Sorry, it'd be nice to have. No, I'm not going to say that. It's not nice. It'd be nice to
Tracy is generous enough and she can come, like, she can join one of your Zoom sessions for 15-20 minutes, not much. Then you can grill her.
Yeah. Would you be willing to do that, Tracy? Or are you?
I can, but I don't know how to create a Zoom meeting. If I receive a link, I can join, but I don't know how to create this.
I know how to create them.
Okay.
Or we could meet together. You guys if you guys will just take I put my number in the chat. If y'all will just text me and then, okay, I'll put down your number. It would just be nice to study with y'all because, yeah.
Mariana is traveling. Poor one, she's actually at airport.
Oh,
she
okay.
managed to jump in, but I think she will be definitely happy to join you, ladies, and do an extra study session. Only thing is you have to reach out to her. I'll ask her if she's okay, and I will pass on your number, Brittany.
Okay, that would be wonderful.
And when you get together, guys, sorry, Brittany?
That would be wonderful. Sorry.
Yeah. When you get together, ladies, not just in my class, any other class, when you get together to meet anybody for study session, doesn't matter they are in the same class or not, when you are studying with anyone, get to the chase. Immediately say, hi, how are you? How is it going? Did you study? Yes, that's all. First 1 minute only for conversation. Immediately jump on what you need to study and get lost. Sorry. I mean to say, use your time well, okay? Just be gone. Don't waste time in anything else, because when you're from the same class, you get into the thing like, oh, did you do that? Oh my God, I missed that. Oh, that homework. Oh, you know, what did he say there? Okay, what did the textbook say? All these unnecessary things come up and you will just simply waste your time. I have done this in my life and I've wasted a lot of time doing study sessions with people. Don't do it. Don't waste your time. Be very mindful of your time, okay? Yeah? It's just my suggestion. Okay, any other questions, ladies? If you text me and I'm not answering them promptly, please don't mind. I'm swamped. I have 10 things that have come up and I'm taking care of them. I'll try to answer, but otherwise take each other's help and email me. I'll try to reply, but don't expect a reply if your test is tomorrow and you're texting me, okay? Bye guys. I have another class coming up.